any kind of deviation from the normal structure or normal number in a chromosome, that is called as chromosomal aberration. In the normal case, when a chromosome is good in shape or good in number, then it is called as euploidy. But when a chromosome deviates from the original structure or from the original number, it is known as aneuploidy. Hello friends, welcome to Biology Concern. I am Animesh. In this video, I am gonna discuss about the chromosomal aberration, that is the structural aberration portion. So let's jump into the topic. First thing, if we consider about euploidy, then euploidy comes from a Greek word, that is eu means good and ploid means fold. That means euploidy defines those chromosomes that are healthy, that are good, or that are good in number or good in shape. And next case, in case of aneuploidy, here an stands for not, u stands for good, just like euploidy, and ploid stands for fold. So when a chromosome deviates from the normal structure, then it is called as aneuploidy. And aneuploidy have two of the division. One is structural and another one is numerical. So one is structural, that means any deviation in structure. So structural means any deviation from normal structure. Okay, and another one is numerical or number. Okay, so what is numerical stands for? Numerical stands for any deviation from normal number. That means normal number of chromosome that should be present inside of a cell. In case of human cell, the normal numbers stands for 46. That means 44 autosomes and 2 sex chromosomes, okay, or allosome. So 44 autosomes and 2 allosomes. Here in this case numerical, if the number from the 46 is deviated, that means either increase or decrease, then those terms as numerical aberration. But today we are going to discuss about only structural portions, that is structural deviation or structural aberration. Structural aberration falls into four major categories, that is deletion, second one is duplication, third one is inversion, and fourth one is translocation. Okay, so today we are going to discuss about four of the major structural aberrations, that is deletion, duplication, inversion, and translocation. So let's jump into those in details. Okay, first we have to consider about deletion. Okay, so what is deletion? So deletion, our first objective is deletion. So what is deletion? Deletion comes from delete. So when a portion of chromosome deleted from its original shape, then it is called as deletion. Okay, so when a part is deleted from the normal chromosome, so when a part of chromosome deleted, then this considered as deletion. Deletion have two subtypes. One is terminal deletion, and second one is internal deletion. Okay, so first thing is terminal and second thing is internal. So what is terminal? So in this case, if we draw a diagram of a chromosome, so this is a submetacentric chromosome, guys. One is Q arm, long arm, and one is P arm, that is the short arm. So if we divide the chromosome into a certain portions, so this loop like that. So this portion is A, B, C, D, E, F, P, Q, R. So this is the easy way to understand a structural portion of the chromosome. This is the centromere. Okay. When in case of terminal deletion, when a segment from the terminal portion is deleted. Okay. So in case of terminal deletion, when a terminal segment, that is the end segment, either this one or this one means A or this one means R. So either a terminal portion deleted. So in case a terminal portion is deleted, so A is deleted from the original structure. So now we have got this structure. Okay, so here B comes the first, C, D, E and F. Here P, Q and R. So in case here A is deleted from the normal chromosomal structure. In case I showed here the A portion, that is the terminal portion of the Q arm. Another portion, that is R portion, that is the terminal portion of the short arm, also be deleted. Okay, so in this case here the short arm will be shorter, but in this case I draw here, in this case the long arm has shorter because the A portion is deleted. So what is internal deletion? Just draw a diagram of chromosome just like previous. Okay, so it's A, B, C, D, E, P, Q, R. So in case here internal means the portion from inside. Okay, so in case here any of the portion except A and R, any of the portion in between A and R, that is B, C, D, E, P, Q, any of the portion, if any of the portion is deleted from the chromosome, then it is called as internal deletion. Okay, so in case here we took an example of C, so the C segment is deleted. Okay, so what we have got now, we have got A, B, D, E and P, Q, R. Okay, so C segment in case here is deleted. So this is the internal deletion. This is about deletion. So jump into the next point, that is the duplication. Okay, so what duplication stands for? Duplication stands for when a portion is duplicated. That means when a portion is copied itself and attached to the same chromosome, this portion is known as duplication, or this phenomenon is known as duplication. Okay, so in case of duplication, we found three of the types of duplication. One is tandem duplication. So first one is tandem duplication. Second one is reverse tandem. Second one is reverse tandem duplication. And third one, that is terminal tandem duplication. Okay, so first one we have got tandem duplication, second one is we have got reverse tandem duplication, and third one we have got a terminal tandem duplication. Before I start about the tandem duplication, let's talk about what is tandem. For the tandem, okay, so what means the tandem? If a segment of a chromosome is repeated one after another and is repeated again and again and again, then those segments are known as tandem segments. In case here, just for example, AC, 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 AC. So adenine cytosine, adenine cytosine, adenine cytosine, adenine cytosine, adenine cytosine, adenine cytosine, and adenine cytosine. In this here, I write it down adenine and cytosine. Okay, repeated again and again and again. So in this case, if a segment, that means A and C, adenine and cytosine, make a segment, and those segment is repeated again and again. Okay, so those segment repeated again and again. In this case, we call this term as tandem. Okay, so what is tandem duplication? So if a part of the chromosome is duplicated and attached next to the original part, then those duplication are known as tandem duplication. So what are they look like? Okay, just draw a diagram of chromosome. So here A, B, C, D, E and P, Q, R. So let's suppose B and C part has been duplicated. Okay, so B and C part has been duplicated and attached just next to the original part. Okay, so in case here we have got A, B, C, B, C, D, E, P, Q and R. So here you have got that B and C portion is duplicated and attached just after the original sequence. Okay, so B and C portion is duplicated, the black portion is duplicated portion, and here B and C, P and C attach just to the original portion, that is the original B and C. So this kind of duplication are known as tandem duplication. In case of reverse tandem duplication, those are same as tandem duplication, but the duplicated portion attach just reversely. Okay, so in this case, if we draw a diagram of chromosome, so A, B, C, D, E, P, Q, R. Okay, so BC portion, let's talk about BC portion. Okay, so BC portion has now been duplicated and attached just reversely to the original segment. So what is look like that? B, C, C, B, B, E, P, Q, R. Okay, so here you will find that the BC portion has been duplicated and it attached just reversely to the next to the original sequence. Okay, so original sequence I mark here in the green color. Okay, so the original sequence is BC and the duplicated portion is BC, but they attach reversely to the next to the original portion. But in case of tandem duplication, the duplicated portion attaches just to the original segment. But here, in case of here reverse tandem, duplicated portion attaches next to the original portion, but in reverse sequence. Okay, here the sequence is C and B, CB, BC, CB. So that is a reverse tandem duplication. But in case of terminal tandem duplication, if a terminal segment duplicated and attached in the terminal segment. Okay, so here just draw a diagram of A, B, C, D, E, P, Q, R. Okay, so let's assume that A and B portion has been now duplicated, and after duplication it attached in the terminal just before the A, just before A. Okay, so in case here we have got AB, AB, C, B, E, P, Q, R. So in case here those AB segment is duplicated. Okay, right? And the original segment is here, A and B. So this is the starting point of the original sequence, A, B, C, D, E, and duplicated portion attached just before to the original portion, that is the terminal sequence. Okay, so when a duplicated portion attached in the terminal sequence, then it is called as terminal tandem. Why it is tandem? Because the sequence are repeated. Okay. In case of inversion, so we have focused on inversion. Okay. So in case of inversion, inversion stands for 180-degree rotation of the original segment. So it is stands for 180-degree rotation of the original segment. Okay. So 180-degree rotation of the original segment. Inversion are classified by into two categories. One is paracentric. Okay. Next one is number two is pericentric. Okay. So what is the major difference between paracentric and pericentric? In case of paracentric, here centromere is not involved. Okay. So without centromere. So in case of paracentric inversion, centromere is not involved. But in case of pericentric, centromere included, right? So in case of pericentric centromere inversion, we have got centromere included. That means centromere involved in the case of inversion of pericentric inversion. But in case of paracentric, here centromere is not involved. So what is that? So if we draw a diagram of chromosome, just like a single thread here. So if we draw a centromere here, so this is the centromere, guys, and this is the segment of A, B, C, D, E, P, Q, R. So what is the about the paracentric? Okay, first thing, the long arm loop, create a loop. Okay, the long arm create a loop. Centromere is here as previous, and P, Q, R portion is there. So the long arm is now looped. Okay, so A, B, C, D, E. So A, B, C, D, E all are there. And now the critical point where the arm is overlapped. Here first make a cut. Okay, so chromosome first makes a cut in here, the joining segment. After that, it produces like that. Okay, so in case here A is here, B is here, C is here, B is here, E is here, P, Q and R. So if we stretch out that particular loop, so we have now got A, B, C, B, E, P, Q and R. In case here, if you observe the original sequence, that is the A, B, C, D, E, and now the inverse sequence, that is DCB, that is the 180-degree rotation of the original sequence. BCD to DCB, 180-degree rotation of the original sequence. So in case here centromere is not involved, and the first a loop created on the long arm, then the joining portion of the loop now splitted, and this segment join with this segment join with this portion, and this segment join with that segment portion, and after that it look like a loop. And then if we stretch out this portion, we have got the new sequence of the our chromosome. Okay, so that is the paracentric inversion. But in case of pericentric inversion, so in case of pericentric inversion, A, B, C, D, E, P, Q, R. So in case here centromere is involved, right? So the first thing is a loop. So centromere is involved here. The loop is formed between long and short arm. Okay, the loop is formed between long and short arm. So what is the sequence? The sequence is A, B, C, D, E, P, Q, R. Okay, now as previous, so P, Q, R. Now, just like previous case, here split occurs in the joining portion. After that, this segment joins with P segment, that means C is joined with P, and Q segment is joined with D segment, right? So after that, we have got a sequence. Okay, here. So A, B, C, P centromere is here, E, D, Q, R. If we stretch out that portion, so we have got now here A, B, C, P centromere is here, and E, D, Q, R. So in case of pericentric inversion, centromere is involved, right? So there is a centromere, there is a centromere, there is a centromere. So long arm converted to short arm and short arm converted to long arm in this here. So both arms are now equal in size. So this is about the pericentric inversion. Now we have looked about the translocation, right? Translocation. So what translocation means for? Translocation actually stands for the exchange of a segment of chromosome, right? The exchange of a segment of a chromosome. Okay. So in case of translocation, so trans means shifting, location means position. Okay. So shifting of position of a certain segment of a chromosome is shifted from one position to another position is known as translocation. Translocation is subdivided into three major categories. One is non-reciprocal intrachromosomal, next one is non-reciprocal interchromosomal, and third one is reciprocal interchromosomal. Okay. So what it stands for non-reciprocal? Non-reciprocal stands for it only gives but not take. Okay, so only give but not take. And intrachromosomal, here you observe the term intrachromosomal and interchromosomal. Intra means within it, that means self, within the self, that is called as intra. But in case of inter, then we have called about two different chromosome. Okay, so in case of inter, the two different chromosome is involved, but in case of intra, that is one chromosome is involved. Let me give you an example. In case of intra, so if we take an example of family, our family, if we call the intra family, that means our within our own family, that means within our parents, within our brothers, sisters, cousins, etc. But if we called about inter family, that means two different families, that neighbors. Okay, neighbors, or a family from here and a family from far distance, so between two families. But in case of intra, that means within it. So in case here, if we draw a diagram of chromosome, so A, B, C, D, E, P, Q, R. So let's assume that the B segment now been translocated, okay, and B segment translocated into between Q and R. Okay, so it is a in same chromosome. So what happened next? So A, C, D, E, P, Q, B, R. So B is now shifted from here to here. Okay, so B is now shifted from here to here, but within the same chromosome, right? But in case of non-reciprocal interchromosomal means the involvement of two chromosomes. If we draw a diagram of two chromosomes, so A, B, C, D, and here M, N, O. And next chromosome is this. So here small a, small b, small c, small d, small p, small q, and small r. Okay, so let's assume that this B section translocated. Okay, and those B section is translocated from chromosome number 1 to chromosome number 2 and is translocated into between C and D portion. So in the next point we have got A, C, D, then M, N, O segment, and in case here in the second chromosome we have got A, B, C, then we have got B, our translocated section, then we have got D, then we have got P, then we have got Q, and then R. So here involvement of two different chromosomes, okay, and one, as I said earlier that non-reciprocal means it only gives, not the take. Okay, so B segment is shift from the chromosome 1 to chromosome 2. Okay, so this is the about non-reciprocal interchromosomal translocation. Let's talk about the reciprocal interchromosomal translocation. Reciprocal means bidirectional. Okay, so bidirectional. So reciprocal means bidirectional, but non-reciprocal means unidirectional. So in case here, if we draw a diagram of chromosome, here A, B, C, D, E, Q, R, and our second chromosome is small a, small b, small c, small d, small p, small q, small r. Okay, so in case here, let's assume that this B segment translocated and it attached just between C and D of first chromosome. Not only that, but this B segment also translocated and it attached the C and D between C and D portion of the second chromosome. So in case here we have got a condition when both of the chromosomes are giving and both of the chromosomes are taking. That means it is a bidirectional relationship. So in case here we have got A, C, next small b, next D, next P, Q. So in case first chromosome we have got A, C, small b, D, P, Q, R. And as because B is shift from here chromosome number 2 to chromosome number 1 and joined with in between C and D of the first chromosome. So in case of second chromosome we have got A, then C, as because B is already shifted. Okay, next those segment B, that segment that is shifted from chromosome number 1 to chromosome number 2, and then D, then centromere, then P, then Q, then R. So in this case both, this is the exchange of bidirectional. Okay, so chromosome number 1 exchange its segment with chromosome number 2. So this is called as reciprocal interchromosomal translocation. So in this video I have covered all of the major structural aberrations occurred in a chromosome: deletion, duplication, inversion, and also translocation. Okay, So I think this video is really helpful for you to understand the basics of the chromosomal aberration, what aberration stands for, what is the deletion, duplication, inversion and translocation stands for. So this is all about, guys. I think this video is really helpful for you. Please hit the like button and don't forget to subscribe. Thanks for watching.